disorders and PhD in physiology and biophysics from Georgetown University. And he completed a residency in pediatrics at the University of Miami, a residency in child neurology, and fellowship in behavioral neurology and learning disabilities at Harvard University Children's Hospital of Boston, and a fellowship in psychology at Boston University. He also has a master's in biomedical science and biostatistics from Drexel University, and has board certifications in pediatric and pediatric neurology. He's authored over 300 peer-reviewed publications and book chapters, and serves on several editorial boards. We are excited for his presentation on treatable folate pathway abnormalities in autism spectrum disorders. And with that, Dr. Frye, why don't you take it away?
Sure. Well, thank you so much. I really appreciate you inviting me to talk about this. It's something that I've been doing for over 20 years now, and I think it helps kids with autism to treat these folate pathways. And really excited to share this with others, so that they help children with autism more. Just some disclosures. We have different funding from foundations, federal government, industry, and several advisory boards. And my disclaimer, that is that I'm going to talk about treatment of folate pathways. None of the treatments that I'll talk about are FDA approved for this, for autism. As we know, there's only 2 approved drugs for autism, which are both antipsychotics, limiting our treatment for any autism symptoms. So I think this is really a very much emerging, area where hopefully we'll have FDA approved drugs in the future. But this being off-label is important for anybody prescribing it to know the risks and benefits, of course, when prescribing medication. So I'm going to talk a little bit about autism from a different point of view. Many people think of it from a behavioral point of view, but we look at it from more of a systems biology medical point of view. That is that we look at autism as a disturbance of the body, both through systemic disorders, including problems with the mitochondria, glutathione metabolism, methylation and epigenetics, and of course genetic disorders, and these affect all the cells in our body. We know that there's organ-based disorders, including the brain. There's many different abnormalities that we can look for, including altering the excitatory inhibitory balance, epileptiform abnormalities, cerebral folate insufficiency, which I'll talk about, but also sleep disruption, which is very important, neurotransmitter abnormalities, autonomic abnormalities, and other things. And of course, we have to remember that GI system symptoms are very prominent and can be very disabling in children with autism, and it's important to take those into account. And we see a lot of immune dysregulation in some kiddos. So we look at it in these kind of a broader sense of what we could do to make the body work better in combination with any behavioral, treatment. And one of the things that is important is these environmental factors. We know that the environment has such an important role. And when I say that, I'm talking about the environment in the broader sense, not things like just toxins or something, but we have so many other environmental factors which we have to really think about, including the psychosocial factors, family stressors, and coping mechanisms. Educational factors, the quality of therapy, the educational setting, the behavioral supports, sensory factors. As we know, many children have sensory sensitivities, which really stop them from being able to really tolerate it can drive symptoms when allergies are worse. And of course, in other environmental toxins like air quality and common chemical toxins like cleaners, and we do see this, that the air quality, can actually affect kids. I know that when there were the fires up in Northern California, all the kiddos with autism, many of them, their behavior was going off the charts, even though they weren't right where the fires were, but just because of the air quality. So it's always important to remember to look at things in the broader sense to optimize function in kiddos with autism. And today I'm going to talk about one carbon metabolism, that is folate metabolism in autism, how important it is and what we can do about it. So first, I'm going to talk about the relationship between folate and one carbon metabolism to methylation and redox abnormalities. So one thing that we know, and this is really outlined by Jill James, who at that time was at Arkansas Children's Hospital. It's kind of an interesting story. She was actually a cancer scientist that studied redox metabolism, and somebody asked her to look into abnormalities in oxidative stress and methylation metabolism in kiddos with neurodevelopmental disabilities, and she came upon the fact that kiddos with autism seem to have abnormalities in many different parts of these pathways. And what we see here, this complicated biochemistry, is we see the transmethylation cycle on the top, which is so important to make SAM, or major methyl donors, which is important for methylating DNA and RNA proteins, also enzymes. And of course, this is in DNA, this is very important with regulation of gene expression. And without being able to turn genes on and off properly, really, it's going to be very difficult to regulate the cell appropriately. And then, transmethylation pours into transsulfuration, which makes glutathione, which we can see at the bottom here is GSH. And GSH, or glutathiones, are major antioxidants in our body. And we know it's depleted in kiddos with autism, which I'll show you in a second. And this is all connected to the folate pathway, which is over here on the left. And the combination, the place where the folate pathway and the methylation pathway comes together is right here at methionine synthase, and B12 is very, very important for this reaction. So it's very important for therapeutics. What we see in autism is very interesting. When Dr. James was looking at this, really discovered that what was happening in a lot of children is that they had low levels of glutathione for some reason down here. And essentially what was happening is the body was trying to make more and more glutathione to support redox systems. And by doing that, it needed more cysteine, the major, the rate limiting amino acid for making glutathione. And that's derived from homocysteine in the methylation cycle. So essentially, to make more glutathione, the the transsulfuration cycle was kind of stealing homocysteine from the methylation cycle and preventing it from really functioning correctly. In this study, it was very interesting because Dr. James, when she found this, and she found that it really seemed to be so significant, she called it an endophenotype or a subset of kids with autism, which we'll see as an understatement in when she first found this. The other thing that she looked at, which was really interesting in the study, was how these different genetic polymorphisms in these enzymes may be affecting the cycle. And of course, we hear so much about the MTHFR as if it's the only gene in the body from many patients. And we have to remember that these are polymorphisms we're looking at that are common changes in the genetic code that in and of themselves could really cause disease because of the fact that they're so common. But Dr. James went in to see if these changes, these polymorphisms, were more prevalent in kiddos with autism, which we know they are. And so here in yellow, I have the genes here that in the cycle that seem to have polymorphisms that were more so affected in kids with autism. But what she did was really interesting, and this really shows one of the problems that we have with genetics is that she found, she didn't look at just single gene polymorphism, but she looked at combinations of polymorphisms. And this is where really the significance comes out, and we can see on the top line here that the polymorphism in the cobalamin carrier and COMT, if you have those two polymorphisms, your odds ratio of having autism is 7. So really that odds ratio is gigantic if you have combinations of polymorphisms. And the same thing, we can see this is the reduced folate carrier and the MTHFR, or the reduced folate carrier and glutathione enzymes, or two MTHFR polymorphisms together. So really the point here is one, the folate pathway and its ability to function does seem to be involved in the risk of autism, which is very important. And two, the genetics is very complicated, you know, and I think that's what we see is that we have really this polygenic, landscape that makes genetics many times difficult to interpret. So this is some of the original data that Dr. James found, was when she compared to controls, kids with autism had abnormalities in, for the most part, every one of those metabolites in the trans methylation and transsulfuration cycle there is. And so it was thought that, well, maybe this does represent a subset of kids with autism. So so she's not the only one that did this research. And there's a good systematic review and meta-analysis in free radical biology and medicine. A little old now, but still it holds up and shows that that multiple groups, so multiple studies by multiple groups throughout the world, replicate this reduction in glutathione and methionine, cysteine elevation in oxidized glutathione, GSSH, and polymorphisms in the MTHFR associated with autism. But not only that, Dr. James' group and others have looked for glutathione abnormalities in many tissues of kids with autism, including the brain. And it's been found, so this isn't something that's just measured in a blood test. It's found in many tissues derived from kiddos with autism. And what does this cause? Well, the redox abnormalities can lead to oxidative damage in DNA, protein, and lipids. And this has been shown over and over in tests. So again, Dr. James thought this was in the phenotype or, a subset of kids with autism. And then she teamed up with Jürgen Hans and Rensselaer Polytechnic Institute up in Troy, New York. And he actually asked, well, if you took these markers, could they be diagnostic of autism? And so using discriminant analysis, he said, well, can we make, discriminant analysis essentially makes a mathematical function that takes these markers to see if it can divide these groups up. And here we see the results. The red dots are the normal, typical participants, and the blue are the ASD participants. And essentially, discriminant analysis found certain markers here that are yellow, which actually distinguished children with autism from those that are typically developing with a 97% accuracy, suggesting that these abnormalities are not just found in a subset of kids with autism, but actually may be diagnostic of autism. And this work is being followed up. Interesting, not only do we have problems with folate, but B12 itself seems to have a problem getting into the brain. And this is a nice study by Richard Deeth, where he actually measures in the brain.
The webinar, and I just get the spinning wheel period.
He actually looked at B12 levels in the brain tissue and found out that they were low. But not only that, these same abnormalities that we see in the blood and other tissues in the body were also found in the brain. And he found different low levels of all types of species of B12. And here we can see on the left here, the same markers, homocysteine, methionine, SAM, SAW, glutathione, were all abnormal for the most part in the brain, or close to abnormal statistically, but mostly statistically significantly abnormal in the brain, as well as kiddos with autism, suggesting that these are just not in the body, but they do extend into the brain. So of course, the question, my question as a doctor is, what can you do about this? So one of the first studies that Dr. James did was look at, is this treatable? And so she used methylcobalamin, that is methyl B12, injected 3 times a week with oral folinic acid, also known as glucoboride, to treat kiddos with autism who had these redox abnormalities. And what we see is, compared to control children, and that many of the glutathione abnormalities, actually these transsulfuration abnormalities, improved significantly with this treatment. And we can see that homocysteine actually increased, which is this an important aspect of this. We usually think of hyperhomocysteinemia in adults, but in kids with autism and other neurodevelopmental disabilities, we actually see low homocysteine And so this treatment actually corrected many of the transsulfuration abnormalities, not so much the transmethylation abnormalities. And we can see here these changes, the glutathione redox ratio in many children increased substantially, and oxidized glutathione decreased substantially in many. We also looked at what type of behavioral changes may have occurred. And so at the beginning and at the end of the study, the Vineland Adaptive Behavior Scale was performed to see what their developmental level was. And I have here in the last column, this change in 3 months of treatment, how many months equivalents, are they, did they change? And what you can see is that, and these are averages, that in language, there was a 6 to 8 month change in language abilities in 3 months. But also in these other important skills, we think are very important in autism, interpersonal skills, play and leisure, coping skills, that some of them, play and leisure skills, on average improve a year in 3 months. So really addressing these abnormalities can be important. There's other ways of treating this. So there's something called N-acetylcysteine. N-acetylcysteine, if you remember from medical school, and of course, if you probably remember from ER medicine, is the antidote for Tylenol. And that's because Tylenol depletes glutathione. N-acetylcysteine is given in medicine for a number of different reasons, but it can actually improve glutathione levels by donating that cysteine, and both improve the production of glutathione and prevent homocysteine from being drained from the methylation cycle. And so this study done by Stanford by Antonio Hardin, where he used N-acetylcysteine 900 milligrams up to 3 times a day in kids with autism, a double-blind placebo-controlled study. His primary outcome measure was irritability, and we can see here very substantially that irritability dropped in those receiving N-acetylcysteine as compared to placebo in the first 4 weeks of treatment. So we can see that this one carbon metabolism biochemical pathways are extremely important. Here I've kind of shown it in a different way, a little bit more organized, I think, to show your methylation pathway on top here, your transsulfuration pathway down here, on the left, your folate cycle. And then the important thing about folate we sometimes don't appreciate, because a lot of people are concentrated on the methylation aspect of it, is that folate's important to making purines, which not only are important for making RNA and DNA, but for making things like GTP, which makes BH4 or tetrahydrobiopterin, which is an important cofactor for making neurotransmitters, dopamine, epinephrine, norepinephrine, serotonin, melatonin, and also nitric oxide metabolism. So these pathways are really essential. So our treatment, as we had talked about it for this, is we give methyl B12 here, which improves the function of methionine synthase, which connects the folate cycle to the methylation cycle. And then we give leucovorin or folinic acid, which comes right into the folate cycle. So, and we'll talk a little bit about folic acid and how it differentiates from reduced folates like leucovorin in a bit. But in this diagram where I have all these red boxes, these are metabolites that have been found to be abnormal over and over and over again in studies, repeated studies, and the circles are your enzymes. Dihydrofolate reductase, which is important for activating folate so it can go into the folate cycle, has polymorphisms that makes it work less efficient. But even with it working efficiently, you only have so much dihydrofolate reductase, so that giving folic acid to treat a folate deficiency is not effective, because the body can only activate so much for its use. And the other thing that's really interesting is that what we believe and what's been shown in several scientific studies, including those that have used magnetic resonance spectroscopy, is that by actually driving this biochemical system to make glutathione, does not just help with redox metabolism, but glutathione is made of cysteine, glycine, and glutamate. And glutamate has been implicated in individuals with autism. That is, the excitability inhibitory ratio of the brain we know is increased, and glutamate is the major excitatory neurotransmitter in the cortex. And this is not limited to autism, but many other psychiatric diseases. So in many diseases, we treat in a similar way, or give N-acetylcysteine, which donates the cysteine to make glutathione, and it actually uses up glutamate and reduces glutamate, or the excitability of the brain, so it actually can balance the neurotransmitters in the brain. So now we'll talk about an area that we've had a lot of success with, and we have ongoing clinical trials, this disruption of folate transport into the brain. So the brain, as we know, is a protected area. Things can't just diffuse into the brain. We have a blood-brain barrier that makes sure that only certain things go into the brain. The brain is a highly controlled environment, and folate is no different. And for folate to get into the brain, it has to go through a complex mechanism using something called the folate receptor alpha, which we can see down here as F. This folate receptor alpha picks up folate, and then it's actually transported across the barrier. And essentially, we can think of it as dumping out folate into the CSF. It's actually more complicated than that, and it does involve actually this folate receptor alpha actually floating in the CSF till it gets to the brain barrier here and releases folate, and then it becomes recycled. So it's very it's a very complicated mechanism to get folate into the brain. And of course, folate is so important, as we talked about. So at about 2004, a little bit before that, about 2002, 2003, Dr. Rainmakers, at that time he's in Germany, he's a pediatric neurologist in Belgium, and he was working with Dr. Blau, who's probably one of the world-renowned best scientists on brain chemistry at the University of Zurich. And he had found that there were some kids he was finding that were not developing, and he didn't know why. He worked them up and he couldn't find the reason why. So he did a lumbar puncture to look at their brain chemistry. And what he found was that many of them had low levels of folate in their brain. And here's one of his first studies, the first 20 patients diagnosed with cerebral folate deficiency. Interestingly, studies that have looked at folate levels over time in kiddos with neurodevelopmental disorders, when you look at it over time, it actually drops in many of these kids and goes lower. And so he reported finally his findings in the New England Journal of Medicine in 2005 in this seminal paper. Now, this paper didn't only introduce cerebral folate deficiency to the world, but the reason why many of the kids were not getting folate into the brain. At first, Dr. Rainmakers thought, well, this must be a genetic problem with the folate receptor alpha. So he sequenced the gene and he could not find one abnormality. At the same time, Dr. Edward Quadros at SUNY Downstate was doing some research on the folate receptor alpha, but in gestation and pregnancy, because it ends up the folate receptor alpha is also on the placenta. And Dr. Quadros found that there is this autoantibody that hung on to the folate receptor alpha and prevented it from functioning. And so in this paper, they introduced what they found was kind of the phenotype of these kiddos. Most of them had neurologic signs starting at about 1 year of age, with decrease in head growth, acquired microcephaly, neurodevelopmental delay and regression, pyramidal tract signs, dyskinesias and movement disorders, and epilepsy in some. And what they found is this went untreated, that sometimes you would have problems with vision and hearing loss. And when they tested them for the folate receptor alpha, as far as titers, they found that yes, indeed, these kids had high levels of this autoantibody that, sorry about that, blocked folate from getting into the nervous system. And so how does this work? So here we have another diagram of the folate receptor alpha here. And it binds onto folate, here represented as MTHF. And it essentially pulls folate into the brain, because the brain's folate level is 2 to 3 times higher than it is in the blood. So this is an active process. The folate receptor antibody, there's 2 of them. There's a binding antibody that binds the folate receptor alpha, or a blocking antibody that blocks folate from binding onto the folate receptor alpha and inhibits its ability to transport folate. Well, what can we do about this? Well, we have an alternative pathway called the reduced folate carrier, which you can see in the middle here. You can see that it's smaller. And the reason it's smaller, it doesn't have the same affinity for folate. And it doesn't actively pull folate into the CSF. So the solution is to give reduced folates, because that's what goes through the reduced folate carrier, at high levels in the blood. So we essentially push folate into the brain to restore the levels. Early on, whenever we had one of these antibodies, we would do an LP to verify that the level of folate was low. But after we studied it and found that the antibodies did indeed correlate with CSF levels, we figured that folate, which was so had such an excellent adverse effect profile, was probably better just to try it rather than undergo lumbar puncture. So as more and more of these studies had described these kiddos with cerebral folate deficiency and the folate autoantibody, they started to describe that a lot of kids had symptoms akin to autism. So at that time, myself and my colleague, Dr. Rosignol, both had autism clinics. So we said, okay, well, how many of the kids with autism are running around our clinic with these antibodies? Is it 5%? Is it 100%? What is it? So we studied the first 93 kids that came into our clinic. We offered the families this test, and we found out that, at least in our clinic, in our population, that 60% of them had the blocking antibody and 50% of them had the binding antibody. So that's a pretty high percentage. 75% of them had one of the two antibodies. Interestingly, these antibodies have also been correlated, these titers of the antibodies have also been correlated with aggressive behavior. And again, when people had studied studied CSF levels of folate, they have found, so this is a study from NIH, from Sue Sweeto, in collaboration with John Schofter and Keith Hyland, where they actually measured the CSF chemistry in kids with autism. And two things they actually found was, one, you can see again, many of them over time, gone untreated, folate levels decreased. And two, just by measuring these levels, 18% of them had low folate levels or cerebral folate deficiency. So again, we can emphasize how important folate is. We have the folate cycle in the middle here. We know that it's important for methylation metabolism, redox metabolism. And over here on the left, we have BH4 metabolism, which is important for monoamine neurotransmitters, nitric oxide production, making RNA and DNA. We know low folate levels results in poor replication of the genome and genetic errors, and also for the mitochondria. So what about giving folic acid? Well, what happens is, again, dihydrofolate reductase can only work so hard. So what happens if you give higher levels of folic acid to treat this? Many become unmetabolized, and we have high levels of unmetabolized folic acid. And we're finding that actually inhibits folate metabolism. And this is really important. Folic acid is the synthetic form of folate. It is not found in nature, so the body cannot use it readily. So what do we do about this? Of course, we use folinic acid or leucovorin, which goes right into the folate cycle and is able to use by the body readily. Leucovorin is really the drug of choice for a number of reasons. And one of the reasons is we have 70 years of experience using leucovorin in oncology to rescue the body from the effects of chemotherapy. So we know about it. We know it's how much to give. We know its side effect profile, and it comes in many forms, including an IV form. So going back to our study of kids with autism, those that had the positive folate receptor antibodies, we offered leucovorin or folinic acid to them at the standard dose of 2 milligrams per kilogram per day, maximum of 50 milligrams per day. And the parents came back in about 4 months and told us what may have improved or gotten worse. We also had a waitlist control group of kids that had testing, and then did not know if they're positive or negative and didn't make any changes. And what we found is that there was significant improvements in receptive language, expressive language, verbal communication, stereotype behavior, and attention. And we can look at it this way, that 2/3 of kiddos had improvements in verbal communication, expressive language, and receptive language. Of course, that's an open label study, so there's biases. So what do? So then we launched the first double-blind placebo-controlled study using leucovorin, or at that time, we described it as high-dose folinic acid. And we found that in 12 weeks, looking at verbal communication measured by either the self or the PLS, depending on the ability of the children, we found significant improvement in language in 12 weeks. We also had all comers, so we could see if the folate receptor antibody actually was predictive. And we found that, yes, indeed, it was predictive of those that was going to respond. And the effect size, 0.91, is large. And the number needed to treat, 1.8, is an excellent effect size, suggesting that probably about 65% of kiddos that you treat will have this response in 3 months. Also, other studies I'll point out have looked at brain chemistry and shown that treating with glucovorin improves methylation, metabolites, SAM, SAW, and homocysteine in the CSF. So since then, there's been other studies, randomized placebo-controlled studies. There's actually 4 other studies. We left out one here from Iran, I believe, that used larger sample size than we did, and all showed significant improvements, and some of them also demonstrating. And they also looked at the folate autoantibodies showing that it is predictive. So it's not just us. Now we have several blinded placebo-controlled studies showing this. So where do we go from here? So right now, we're taking this the next step. We have ongoing trials looking at levoglucovorin. Of course, every drug is made of 2 isomers, the L isomer and the T isomer. And here we use the L isomer, which is the only the active form of glucovorin. In addition, it's a very safe formulation with minimal additives. And this I cannot emphasize enough. Commercial forms of leucovorin have additives, not only dairy and milk derivatives, which children are sensitive to, but many dyes and other incipients. And this is not theoretical. We have had kids regress on certain brands of leucovorin and found that it's because they have many incipients in this, and it's not just 1 or 2 children. It's over and over again, we hear the story. So it's very important that we have a specific leucovorin made for children with autism that is high quality without additives that will disrupt their abilities. So right now we're using a product from Apifo, which is levoglucovorin drop. It's actually tasteless and can be put in water and not detected by children. It's very easy to give. And it has a microdrop delivery system, so it can really be accurately measured. There is 3 trials, multi-center trials, that are looking at this. The NIH one, which was what we call the school age children study from children from 5 to 17, has completed. And the two other studies, one from Autism Speaks and the Department of Defense, we're doing in collaboration with Dr. Huberman at SUNY Downstate. And that should be finished up by the end of the year. So we have some good data on levo before it. So now we can extend this further. So not only is this important in children with autism, but as I briefly mentioned before, the folate receptor alpha is not just on the blood-brain barrier, but it's on the maternal placental interface. And what we find is that about half of the mothers of kids with autism with a folate autoantibody also have the antibody themselves. So some kids may have had what we call a double whammy, and they might have had problems with getting folate to the fetus, to themselves when they were growing. And so Dr. Quatros at SUNY Downstate has developed an animal model showing that indeed, if you inject the pregnant rats, the dams, with antibodies, folate receptor alpha antibodies, that the pups will come out with cognitive and behavioral abnormalities that is not unlike autism. Most importantly, he showed that either dexamethasone or folinic acid given to the mother that was injected with these antibodies rescues the pups from these behavioral abnormalities. One study that Dr. Quatros performed along with the group in Staten Island looked at the prevalence of these antibodies in the families and found that the folate receptor antibody seems to run in families and is seen in both the father, the mother, and the sibling. And right now we're continuing this with a study where we're looking at the transgenerational effect of this folate autoantibody. Dr. Rainmakers came up from his clinical experience with this idea that depending on whether the mother, the father, or both have the folate autoantibody, you may have different severities of autism, which may or may not respond to treatment. So I'd like to thank you for your attention. I hope that was informative and you can take that into your clinical practice and help some kiddos.
Thank you so much, Dr. Frye. We do have a lot of questions in the chat, but I do want to allow Dr. Huberman, because we do have a lot of local developmental behavioral pediatricians in this session. And so, Dr. Huberman, if you could just take 2 minutes to just talk about the study that you're doing at SUNY Downstate and how families can enroll. And I'm just going to drop the link and the phone number that you shared with us for everyone in the chat box while you're talking. So I can unmute you, or I'm asking you to unmute. Okay, go ahead, Dr. Huberman. I think you're unmuted now. I think you're unmuted, but I can't hear anything, Dr. Huberman, so I don't know if you're trying to speak. But we can't hear you. I know you had some other people on your team that are here. I don't know if one of them can unmute to speak. I'll unmute Victoria, or ask to unmute.
Hi.
Hi, we can hear you.
Yeah, sorry. I'm not sure where Dr. Huberman is.
He's like having trouble speaking.
Yeah, I think so too. All right,
If you don't have a summary of how we can enroll, I'll just put in the information.
I do. Why not?
Okay, go ahead.
Okay, I was just trying to see if I could get in touch with Dr. Huberman, but how we can enroll, we have an email that interested families can reach out to us on. We also have a phone number. It depends on a lot of criteria because we have a screening protocol that we go through. So what we do would be to get a little bit of background from the interested family. We also enroll depending on the age of the child, because some of the studies have the age eligibility criteria. So for the leukovariant study, we have an age eligibility criteria from 2.5 to about 5 and 6 months. So if they qualify for that, we also make sure like they don't have some comorbidities like epilepsy and genetic malformations, brain malformations. So that's some of the eligibility criteria that we go through, and based off that, once we kind of look at the developmental history, we get in touch with the families and register them or enroll them into the study, and then they can come in for the first screening visit. We also have the other study called the transgenerational study, which we collect specimens or lab specimens from the biological parents and also the children of the parents. We do that. It's a minimum of 1 in-person visit to Brooklyn downstate. Then they could potentially give other samples like our saliva, urine, and hair. We have a variety of things that could be given, but the minimum requirement is a blood specimen. Then we also have another study, the Brain Foundation study, and that would be looking at pregnant persons as well as the development of the newborns. So we collect blood before the delivery process, and during delivery we collect cord blood, and we follow the newborn till a year after birth. Also collecting blood just to look at the time of development of the folate receptor autoantibodies. So in terms of enrollment, we kind of give more of an insight into these when the family reach out to us. So would you want me to just drop the number here?
I think I put the number in the chat for everyone. But I'm gonna be sending up a follow-up email with the recording. And so certainly if you have a handout that you use to enroll families with that information summarized, you can certainly pass that along to me and I can include that in the follow-up email.
Have flyers and brochures, so I'll be happy to send that your way.
Okay.
Yeah, I think
Okay.
that we get to you. Yeah, sure.
Okay, that sounds good.
Yeah.
Dr. Huberman, I don't know if you're able to speak now. I see your video. You're not muted. So, but I don't think we can hear you still, unfortunately. Yeah, you can, if you want to add anything else in the chat, you can. I'm gonna switch to questions because we have a lot. One that was recurring, Dr. Frye, was whether we could also get a copy of your slides to send out. Yes, okay, all right, so we will be sending that out in a follow-up email. So if you got an email with the invitation, you'll get the follow-up email with a link to the recording and Dr. Frye's slides. Let's go to some of the questions that people have been asking. So one question that came up is these folate receptor antibodies. How often are they seen in kids without autism? Because like MTHFR is very common in kids without autism, so it's sort of hard to interpret the clinical significance.
So, yeah, so we'll go back here and through it quickly. So this is the study from from Dr. Quatros and Staten Island group here. And so we see, it looks like you see about 25-30% of the time. They're definitely at lower titers too. It's not only the positive, negative. Other studies have found it at lower amounts. We did a meta-analysis a couple of years ago, where we looked at this and combined, you know, all of the studies. That's, I'll send you that too, because that has a lot of good information in it. As far as really overall what the literature has shown. This is actually before a lot of these other studies were done, so really we need a kind of almost another meta-analysis now to take in those other studies. But let's see. So in typically developing non-siblings, our meta-analysis found that either one was 15%, and with the blocking being 4% and the binding being 10%. Dr. Rainmakers did a study where he looked at it in developmentally delayed children without autism. He found that it was 5%. Whatever transgenerational study, one of the things that it's based on is the fact that that typically developing siblings of kids with autism have a high rate of this antibody, about 61%. So the question is, why don't they develop autism? And I think the answer there goes back to my original, how I started out with the fact it's complicated. You know, it has multiple factors. And one of the things we're doing in the transgenerational study is we're matching those typically developing siblings and measuring many other factors, including genetic polymorphisms, to see what that other factor is that pushes them over the ledge to cause them to have autism. So as the answer to most science is, it's complicated.
Yeah. We can see that. I do want to be mindful of time because we are at the 11 o'clock hour. I'm going to stop the recording here. But Dr. Frye, if you're able to continue staying on to continue answering questions, we'd love to have you. I'm not sure what your time availability is.
I put in some time, about a half hour for that. If
Okay.
do that.
Perfect. So I'm just going to stop the recording here.